I'm Professor Matthew Schmidt, and I'd like to welcome you to Genetics. In this section, we're going to discuss once again linkage and gene mapping, but specifically we're going to talk about the way we can do this with certain types of fungi, and we can simplify the analysis on the one hand, and we can do some things that you really can't do any other way. So who knew fungi were so important in genetics? You do. So let's take a look at the concept of I could be wrong, but a lot of people find themselves a little bit in the dark when it comes to fungal life cycles and genetics and things like that. They are different in certain ways than animals and plants, and I mean not just the obvious ways. So certain fungi that are technically in the division Ascomycota, um, they're in that division because an ascus is a little sac in which they make their sexually generated spores. So they make these sacs of either 4 to 8 spores, and we can use them for a particularly powerful type of genetic mapping. It's called tetrad analysis. And we'll see more about tetrads in a minute and why this is so powerful. What a tetrad is, perhaps obviously, we've used that term before, but it's a little different here. Tetrad generally means 4, but there are 4 spores in one ascus, and it's not a coincidence because remember, in meiosis, generally speaking, one starting cell leads to 4 haploid gametes. In this case, they're not really gametes because of aspects of the fungal life cycle that we'll discuss very briefly, but what they are spores, and there are 4 spores in one ascus, and this is very important. That represents the product of one single meiosis event. In that little ascus, that little sac, the 4 spores that are in there came from one meiosis event. So just think for 1 second about what we've been doing. If we do mapping, say in Drosophila, we're actually counting offspring, right? And those offspring are representative of certain combinations of gametes going together. But the reason the numbers never match exactly of like the parental classes, etc., is because, you know, meiosis is going on in hundreds, if not thousands and millions of cells, and I mean it depends on the organism, but ridiculous amounts of meiosis is going on, and then all the gametes sort of end up in one pool, and they randomly sort of connect with another gamete. So we're getting an estimate, which is fine, but here we actually can tell, gee, those 4 cells, spores, came from one meiosis event. It may not be obvious why that's powerful right now, but it will be in a minute. So Neurospora is a mold, and the word mold is a little odd because technically it doesn't really mean that much, but Neurospora is famous in genetics because you get this ascus, but then an additional mitotic division occurs. This is probably just to make more spores numerically so that the fungus can be more prolific. But so technically this is called an octad, which means 8 spores, but really think of them as 4 because there's sort of 2 identical ones and then the next 2 identical ones, etc. What's very important about Neurospora is that they are actually ordered. Now, there's a lot of technicalities that go on with this. And by the way, your class may or may not even broach this subject. Some people like fungal mapping and fungal genetics. Some people shy away from it. So obviously you'll only be watching this if it's relevant to you. But the bottom line here is if you have ordered spores in an octad, you can do something called centromere mapping, and there's really no other good way to do it. So that's something I'd like to explore, and if it's something you have to do, I guess you'll be glad that we explored it. Okay? So why is this so amazing? Well, a couple of things. If you can, instead of counting offspring, for example, like we do in fruit flies, you can actually look at the spores directly inside of an ascus, and you can make sometimes direct observations of spore phenotypes. Now, what might they be? The spores could be different colors, they could be different shapes. It doesn't really matter for our purposes. But one interesting thing is that spores are haploid, right? They're the product of meiosis. So since they are haploid, the genotype equals the phenotype. We don't have to worry about any dominance effects or things like that. So not only, so I have a picture down here where I made it up, but big A would equal a yellow color, little a would equal a green color, and you'd be able to look right under the microscope at that and see that that was the case for those 4 spores that came from one meiosis event. Okay? Now, we're definitely not going to get into this next part. It's rather technical. I just want you to be aware in case your class mentions it. The fact that meiosis occurs during the 4-strand stage of meiosis, where the tetrads are, we've talked about the fact that it does occur that way. The way it was actually proven that that was the case was using fungal genetics and actually using this type of analysis with the tetrads and the spores. All right, so I don't want to say don't worry about it. It depends strongly on your professor. It's beyond the scope of this discussion to talk about that, but that was the evidence that really was the proof, the strong evidence that meiosis occurs at the 4-strand stage. Before we can do this, I think it's a good idea, before we can see how we can use this for mapping, we should take a little look at the generalized life cycle of a fungus, because it really is a bit odd compared with even plants. A lot of people think fungi and plants are similar, but they really have very little to do with one another. So here's a rather beautiful picture, I think. And since, it's a life cycle, we can start anywhere, but why don't we start here, because we just had a similar picture there. That's an ascus that has 4 spores in it. 2 happen to be capital A, 2 happen to be small a. Right now, it really doesn't matter what those 2 things mean. But a spore is something, if this was Neurospora, right, you'd get this mitotic division, and I didn't bother writing the letters of the alleles in there, but now we have 8 spores in the ascus, right? Now, the whole point of a spore, the word spore, I don't want to go into it crazily, but it's used in a little bit of a different sense, whether you're talking about fungi or bacteria or plants. In this case, the point of a fungal spore is basically to either fly in the wind or float on the water, and basically it's going to find a nice place and germinate. So in most cases, you have these haploid spores that have been released out into the environment, and when they germinate, most fungi are multicellular creatures, and the product of that germination is something we wrote here. It's the mycelium is the body of the fungus, if you could think of it that way. But each of these little strands is called a hypha. All right? A hypha is literally, sorry I can't show you one, but it's like a hair in terms of its width, and can grow very, very long. So obviously it has nothing to do with the human hair really, but that's about the width that we're talking about. So one of those strands is called a hypha. The net sum of all the hypha are called the mycelium, which is the fungal body. Okay? Now, there are different mating types in fungi, and in the most simple version, it's a little bit of a stretch to call them male and female. But it's sort of, that's the idea, that if a mycelium that's of one mating type sort of literally runs into a mycelium or a hypha of another mating type, those cells can fuse, and since they're both haploid, this is a little odd. Note that the spore germinated and it was still haploid, and all this is all haploid. Fungi are haploid dominant creatures, which is the opposite of most plants and animals. So if 2 compatible strands come together, they can fuse, and what you end up with is a diploid zygote. Now, I just drew it as if it's one cell. Sometimes it is one cell where the 2 hyphae come together. You might see like the rest of the hyphae going on either side of that. I know this isn't a mycology class, which is the study of fungi, but I just want you to understand. So then, for most fungi, the only time they're diploid is at this stage right here, and then meiosis occurs right away, and you go back to having these four spores, all right? A sexual life cycle, guys, remember, you have to have alternation of generations, but nobody said exactly when you have to have it. So fungi have a little different strategy than do plants or animals. So generally, though, what we need to focus on is the event that has a diploid cell undergoing meiosis and producing spores. I also thought it would be interesting just to get an idea, not all fungi, but many of them are either saprobes or saprophytes, which means eaters of the dead, which is a little bit creepy, but in other words, they're decomposers. Some of them are pathogenic on plants, as the one you see here. It grows through, in this case, a leaf and actually sort of sucks the nutrients out of the leaf. And then here you see an ascus with ascospores in there. This would be something like a Neurospora situation. And a lot of plants, if you see them not looking so great, often it might be because of a fungal infection. But this is just to give you an idea of how the fungus grows. So in other words, this is one hypha, this is another hypha. If you could see all of them together, we'd call it a mycelium. Some mycelia can be ridiculously gigantic, sometimes under the ground in the soil. Anyway, just for the record, a lot of times we'll talk about yeast, or your class may discuss them. Yeast are sort of unique among fungi because they are unicellular, generally speaking. But I want you to see that the same principle applies. So yeast are in the division Ascomycota. They make a nice ascus here. I used A and alpha as the two different loci here because those are the mating type loci in yeast, so you can keep track of them just as well as anything else. But my point is here, so you have spores that are alpha and spores that are A. Germination in the case of yeast, for the sake of argument, let's just say they really don't grow hypha. They stay single-celled. So you have this little situation where they tend to have this vegetative life cycle, which means that they can asexually reproduce for a while. I mean, they can do it. Eventually, and this one over here is asexually reproducing, but eventually an A cell meets up with an alpha cell, and they form a diploid zygote. It doesn't always happen, but sometimes even that zygote that's diploid can go on ahead and reproduce itself asexually. But the whole point of a sexual life cycle, it's got to happen sometime to create that diversity. So when this zygote, it's very similar to the other situation, is ready to undergo meiosis, the result is an ascus that has 4 spores in it. So there's really not a mycelium in play here, but it's the same idea. And yeast are very commonly used in genetics as well as in, well, baking and a lot of other things, right? But they do have a similar type of life cycle and lifestyle as other fungi. All right, let's get down to how we actually do mapping with these guys. Tetrads. So in yeast, it happens to be the fact that we get unordered tetrads. Whether they're ordered or unordered, it doesn't matter for a certain type of mapping that's rather popular. It's useful in its own way. In Neurospora, just for the record, the tetrads, remember we said they're octads, they're ordered. And that means that the order of the spores literally reflects the arrangement and segregation or separation of the actual chromosomes during one meiosis event. What does this allow us to do? I asked the question, now I'm going to answer it. This allows us to do the centromere mapping that I mentioned at the beginning. So that's really the only way to do centromere mapping. If that's something your professor seems keen on, it's definitely a good one to learn, obviously. Okay, how do we do it? So, guys, before you even look at this, please, the idea of mapping is always the same. We want to know how many recombinants we have and compare that to the number of total, right? And that implies, just like we did before, a crossover frequency, etc. But it's going to seem different now. I'll try and keep the two concepts together. It's going to seem different, and this is actually easier to do because we're going to use tetrad analysis to map genes using asci, which is the plural of ascus. All right? So let's do a simple cross involving two linked genes. And the cross is going to be AB, or small a, small b, times, we're using the terminology like flies, the wild type, right? A plus B plus. So when you cross those, remember, this is interesting and a little different to start out with because, it's hard to call these parents, but these are the initial haploid cells that are coming together. They're fusing to make a diploid zygote, right? And that zygote is going to do meiosis. Now, as that happens, look back and think, chromosomally it's happening just the same as it did in the flies. So, but we want to classify each ascus as a certain type. So let's just understand what the types mean, then we can see how they formed and why they allow us to be able to make a map. A parental ditype is named because ditype means that there are only two types of spores in there, and in this case those spores resemble the parents, even though I know they're not parents in the traditional sense, but they were the original cells that generated this, right? So a parental ditype means here's an ascus with two types of spores in it, and they look just like the parents. Perhaps, obviously, a non-parental ditype also has two types of spores in it, but they are absolutely not. They're the opposite of the parent, right? A plus B and AB plus are recombinants, so they're non-parental. We once said that non-parental pretty much means recombinant. And then there's a tetratype, which you could probably figure out means there are 4 different types of spores in there, all the possible combinations. 2 that are parental, 2 that are recombinant, all right? So think of it how it could come to be this way. And right now, for the record, let's think about linked genes, all right? A and B. So remember, we drew this once before, and we wanted to emphasize that when crossing over actually does occur, it occurs at the stage that's a tetrad. And what I want you to see from this is, if no crossing over occurs at all, right, then little a and little b are going to travel together. A plus and B plus are going to travel together. And what that means is they're going to end up right there in that ascus. Only the 2 types, the parental types. So when you see a parental ditype, that means that no crossing over occurred. To make that type of an ascus, all those are parental types. Okay? Now let's think about, use the same drawing, just erase a little bit of this. What if a single crossover occurs? And remember we said, if it does occur, it involves 2 non-sister chromatids, right? So can you see what's going to happen is, this is going to create the tetratype on the other side there. I'm sorry to draw lines all over the place, but this one, remember, it didn't get touched. That's the AB. This one down here didn't get touched. But the 2 in the middle that exchanged a portion, this one is now going to be A plus B, right there, right? This one is going to be A, B plus. So a tetratype ascus is the result of a single crossover event between 2 loci. Now, I'm going to keep that picture and just add to it, because non-parental ditypes are the result of a double crossover, but be very careful that you understand what I mean. What we just drew here is a single crossover event, right, between 2 non-sister chromatids. We've mentioned before, and you guys know it's true, it's also possible that the other 2 non-sister chromatids could engage in a crossover event. So in that sense, it would be a double crossover involving all 4 of the chromatids at that 4-strand stage. And now we're going to get another A plus and B, and another A and B plus. That's why look before, we had two recombinants. Well, we have two recombinants again, but now we have two more because that same crossover happened again with the other strands. So the bottom line is a non-parental ditype represents every single one of those spores in there, all four of them are recombinant ones. So this is the best way to think of it. In a parental ditype, there are no recombinants. Oops, sorry, it's bleeding over into the non-parentals. In non-parental ditypes, all the spores are recombinant because of the double crossover. In the tetratype, we know it's the result of one crossover, but what does that mean? It means two of the spores are recombinant and two of them are not. They're parental, right? So with the tetratype, 1/2 or 2 out of the 4 are recombinant. And if this happened to be an octad, it will be 4 out of the 8. But just looking at this, it looks like a lot of craziness going on. But that's the bottom line. The difference here is we're always trying to get at the recombinants, but now we can say, look, instead of counting the actual offspring or even the spores, let's say if I see one parental ditype, I know all those spores not recombinant. If I see a non-parental ditype, I know they're all recombinant. And if I see a tetratype, I know half of those spores are recombinant. So let's actually put that knowledge into action. So, oh, by the way, I'm sorry, I wanted to mention one more thing. If genes were not linked and they're independently assorting, we should get, this is so important, relatively equal numbers of NPDs and PDs, because an NPD would simply be the result of independent assortment. Hopefully that makes sense to you. An NPD where you basically have 4 spores, none of which look like the parent, that's going to happen about half the time, and about half the time you're going to have parental ones. Okay, so I'm going to draw this down here a little. There's a rule 1 here, but the rule can come in 2 flavors. If parental ditypes, I want to say equal, but they don't have to exactly equal. If they're about found in the same number as NPDs, you're going to assume that the genes are unlinked, meaning that they're independently assorting. However, as I wrote here, if the number of parental ditypes is far greater than the number of NPDs, that's a very, very strong indication that the genes are linked. Because, right, if they're linked, crossing over occurs a certain amount of times, but this occurs from a double crossover, which is even rarer, so you're likely to see a lot less NPDs than PDs. All right? So here's an example of a map for linked genes, and I drew them out, but here's data that you could get. They could say, even if they tell you the cross or they don't tell you the cross, you see 88, 10, and 2. The parental ditypes will always be in the most amount, the tetratypes will be next, and the NPDs will be the least prevalent. So basically, we want to recognize that we're counting 88 PDs in this case, but all the spores that are in there are parental. We're counting 2 NPDs, all the spores in them are recombinant. And we're counting 10 tetratypes. Remember, only 2, half of the spores in the tetratype are actually recombinant. So how do we use that in a formula? Sometimes you may have to memorize this formula, sometimes you might not. But again, if you conceptually understand it, the formula is almost irrelevant. Here's the formula for map distance, and I kept the data up on top just so we can refer to it. It's the number of non-parental ditypes, NPDs, plus T over 2, which really means half the number of tetratypes divided by the total. So let's just do the math, and then we can explain why this is the case if it's not obvious. Number of non-parental ditypes, we counted 2, right? T over 2, so we had 10 tetratypes. 10 divided by 2 is 5, so we're going to add 5 over there. And I made the math easy just in the sense that if you add them all up, you'll see there are 100 total asci that we were counting. So 2 plus 5 equals 7 out of the total of 100 gives us 0.07 or 7%. And this is exactly the same as before. We just got at it a different way. 7% crossing over means that 7% of the time a crossover event occurred, and then we're going to draw a map just like we did before and say that A and B, the loci A and B, are separated by 7 map units, as evidenced by the fact that 7% of the time we get recombinant spores in this case from them. But look what we did. We did it easy. We didn't have to count every single spore. We counted the asci, right? So why does this work? It works because we're counting all the NPDs because they represent all recombinant spores. We're counting the tetratypes but dividing it by 2 because only 2 of the spores there are recombinant. The other 2 are parental. So what we're really interested in is the number of recombinant spores. Even though we want to make an easier way to do it, we have to divide by 2 to get that. And the parentals, right? No crossing over happened there. Remember when we did it with the flies, all we did was said we want the number of recombinants over the total. So we didn't actually count parentals per se. We counted recombinants and divided it by the total. So that's one way to do it. With unordered or ordered tetrads, you identify parental ditypes, NPDs, and tetratypes, and use this formula right here. Okay? I think it's not so bad. I hope it's not. Now, one other thing you can do is centromere mapping. I like centromere mapping. You may not share my affection, but you may have to do it. And pretty much if you do this, you're going to be using ordered octads, probably from Neurospora. It's really the only method by which the gene's relationship to the centromere can be mapped. And if you want to do a problem about this, there's one of the problem-solving sessions that you can challenge yourself with. All right? Now, what is the centromere again? Remember, the centromere is the central portion of the chromosome that interacts with the spindle fibers during cell division. That's the place where the microtubules hook on and pull, right? As such, the centromere really doesn't have a phenotype. Like, you can't say, oh, this one has this type of centromere, this one has this. It's a thing, it's a place. This is why it's challenging, but it is, you could imagine, also useful to know a gene's position with respect to the centromere, and this is the only way to figure that out. So, just to reiterate, the reason this is challenging is because the centromere has no phenotype. There's not like big centromere, little centromere. You can see them in the offspring. Every chromosome has a centromere, but they're the same in every one. So, if you wanted to ask the question, how far in map units is a gene locus from the centromere, you're going to have to do it a very, very special way. So, just because I picked it, let's use the mating type locus. And the 2 alleles for that are A and alpha. We saw that in yeast, but it's true in Neurospora as well. So remember, we're starting off with a haploid A times an alpha. We're going to make this diploid cell that is A alpha, and it's going to right away undergo meiosis. And you'll see in the following slide, but what's going to happen is we're going to get ordered octads that literally show the way the chromosomes separated during one meiotic event. So if no crossing over occurs, we're going to first see what that is going to look like. Then we'll see how we can tell if crossing over occurs between the centromere and a locus. And that's what we would need to count in order to find out a map distance. Okay? Now, I drew this picture, and I apologize that it's a tiny bit askew. I drew it on a Macintosh, and those of you watching me on a Mac who are enlightened, no offense, PC users, I like Macs. But unfortunately, when I transferred it to the PC, it got a little weird. That happens sometimes. I apologize, but I think it's fine. And mainly the point here is to understand what's going on. So let's just follow along. This is important. If no crossovers occur, here I drew a chromosome where we're looking at the mating type locus, which is right here, and the centromere is right here. It's just a little ball, is the way I've drawn it. So we really ultimately want to get at is what's the distance between there and there in map units. So when I say if no crossover occurs, I'm talking about no crossovers occurring in that interval between the centromere and between the locus. All right? So you guys know meiosis. What's going to happen? Meiosis 1 separates the whole homologous pair, right? So now we've got one cell that still, it's haploid already, but it's still AA, and the other cell is haploid, but it's alpha alpha, right? And then what happens in meiosis 2? The sister chromatids separate, right? And so after the mitosis happens, we're going to have an ascus that basically has 4 A's right in a row and 4 alphas right in a row. Remember, because they're ordered. This is literally, looking at this ascus allows you to envision what occurred during this one meiosis. And if you see this, now the way to look at this, if it's an octet, is look at the 4 on the top or the 4 on the bottom. If they are all identical, that means this is what they call M1, or a first division segregation pattern. That sounds fancy. All it means is that the A allele and the alpha allele were separated or segregated after meiosis, the first meiotic division. When meiosis 1 was complete, they were separated. What it also means is that no crossovers occurred. So if you see an ascus that looks like this, you call it an M1 situation, and you know no crossovers happened. That doesn't help us in mapping, though, because in essence, these are all parental. If you know what I mean, I hope. You can't say that they look like the parents because, again, the centromere has no phenotype, but no crossover occurred, so these are the same chromosomes that went right through without any tampering from the original parents. But what if there is a crossover between the centromere and the locus? So in this picture, it looks sort of similar, but this is if a crossover does occur. I animated it. I hope I did a good job. So it's a little crudely drawn, but this is what I'm talking about. What if a crossover occurs where that X is between the centromere and the mating type locus? Look what's going to occur. On the one hand, drawn in yellow here, I hope you can see that, but I'll do a little more. Attached to this centromere right there, we now are going to have this. We're going to have A. And attached to, remember, when a crossover happens, there's always one side and the other side. So in red here, attached to this centromere now, we're going to have the alpha. If you disentangle this all, guys, you can see it. Look, this centromere is what's being pulled over in meiosis, right? And right now it has one chromatid that has an A on it, and it has one that has an alpha. And that's why you get this. And the same goes for this cell. This is the centromere that's being pulled over here, and it has an alpha on it, but now it also has that A on it. It's not that big a mystery. In meiosis 2, the sister chromatids get pulled apart, right? And after the mitotic division that makes 8, look what you have now. If you look at the top or bottom 4, you're going to see that they're not identical. Some are A, some are alpha. Some are A, some are alpha. That is what's referred to as a M2, or a second division segregation pattern. Again, all you have to do is look at either the top 4 or the bottom 4. If they're not the same, that's M2. Why is it second division? Because after meiosis 1 was done, A and alpha were still together. They didn't separate from each other until meiosis 2 because of the crossover. Okay? So when you see an M2, what that implies is that a single crossover event occurred between the centromere and the locus of interest. So then all we have to do is make a map. Oh, I'm sorry, this is a really, really good picture that we made. I thought it wasn't amenable to the teaching of it, but you can follow through what's going on here. So it shows you how a first division segregation pattern comes about, and that's with no crossovers, right? So you're following this through meiosis 1 and meiosis 2. It's very similar to what we had, just drawn a little bit more professionally. But the bottom line is here. Where they put that line, if you look at the top, in this case it was color. If you see some brown colored spores, I almost said beans, they look like beans to me. Brown colored spores, all 4 of them the same, or beige colored, that's M1 segregation pattern. It means no crossover happened in this situation. Here you see right there a crossover is occurring. And I like the way they do it. Now you can see that, not only the allele was changed, but basically a whole segment of the chromosome was traded here. And, if you need to pause this and look, it's really what we just did. You have to follow it through. But the bottom line is, if always draw that line so that 4 are above and 4 are below. If they're not the same, that's an M2, and that means that a crossover occurred. Look, there's all different ways it can happen depending on exactly which sister chromatids are involved in the crossover. But at the risk of redundancy, if there are 4 in a row and they're not the same, you know that that's an M2 pattern. Here, we've said before, a cell doesn't know right side up, upside down. These 2 things really are the same thing. If I turn one upside down, it's going to look like the other. Okay? So how do we draw our map based on this? So here's some data. And what often will be asked of you is they'll say, here's some data. Go ahead, map the centromere. And you start to panic and say, but what are you talking about? I don't know. But it's not bad, guys. I don't want to say it's easy. I know that's annoying and it sounds condescending. It wasn't easy for me when I learned it. But the only thing you have to do is say, all right, don't panic. Just identify who's M1 and who's M2. All right? So if you want, if they were all lined up like this, if I could draw better, you just make this line that separates the top 4 from the bottom 4. And ask yourself only this question. Are they all the same or are they not? So in this case, they're all the same. So this is an M1, right? This is upside down version of that. That's also an M1. Take your time and look, but in all of these, you're going to see that they're not the same, which means it's an M2. M2. Remember we just saw a picture that had all different forms, as it were, of M2? So guys, I hope 2 things that I hope make sense to you is, number 1, as we always see, the parentals, if you will, which in this case M1 is like a parental because no crossover occurred. They're present in greater abundance, right? The M2s, where a crossover did occur, are present in less abundance. What is it that we do whenever we want to map a gene? We say, look, how many recombinants are there divided by the total? So that's what we're going to do here, but you got to be a little bit careful. The formula is this. The percentage of M2 asci divided by 2. I'll explain why that is in a second. Some of you may already see it. But first do the math. So this is the only one we did. We're saying take the percentage of M2. You would almost think that is the crossover frequency, right? Since M2's had a crossover in it. So let's add them up. M2, 9 plus 11 is 20. Plus 10 is 30, plus 12 is 42. So there are 42 total M2s. The M1s, 126 and 132, are 258 M1s. So the grand total is 300. And again, I did that just to make the math a little bit easier. So the percent of M2s is what? It's 42 divided by 300. And if you do that math, you'll find out that it's 14% of the total amount of asci where M2 we're exhibiting the M2 pattern. Now, so there it is, 14%. Why do you have to divide it by 2, you guys? Some of you may be seeing this, but if you don't, look here. These M2, in a way, they're like a tetratype in the following sense. One crossover occurred, right? It may not be obvious just to look at it here, but what that means is that in fact, and if you go back and look, you'll see this, that half of the spores in there are recombinant, but the other half did not get touched. In other words, a crossover occurred between 2 non-sister chromatids, but there were the other 2 in the tetrad there that didn't get touched at all. So counting the M2s is great, but you have to realize that only half the spores in there are actually recombinant. That's why we do the dividing by 2. This formula you can memorize, but it really is just common sense if you understand it. So we had 14% M2s, divide that by 2, that gives us 7% all we're trying to get at is the recombinants, and that's what it is. So now we have 7 map units between the centromere and the A alpha locus. I say that because either allele could be, at that location. 7 map units. It's fun, believe me. In case you didn't know, it is. One last thing, as if that wasn't enough. Oh, I'm sorry. I had animated the M1s and the M2s, but we drew them in, so it's okay. Say you were doing, and I believe there's a question that's a little challenging in the problem solving. You can do this not only with mapping 2 loci with each other and one with the centromere. You can have one where there's the A alpha locus, for example, the B locus, and the centromere, and you need to make a map. It's almost like a 3-point cross, but you have to do it using, NPDs and M1s, etc. I just want you to see this. The same principle applies in that if you calculated like we just did, 7 map units between the centromere and the A alpha locus, but then you had other information independently, you found out through the NPD PD tetratype method that B and A loci were 5 map units apart, and then by the method we just used you found out that B and the centromere were separated by 4 map units. The smaller distances are still more accurate, and it's for the same reason. It's because you're missing out on some of the crossovers, because sometimes there are the other type of double crossovers that regenerate the parentals. Bottom line is, always use the smaller distances if you want accuracy. So the more accurate map distance between the centromere and the A alpha locus, if you have all this information, is definitely 9 map units, even though that's more than... Remember, your calculation is almost always an underestimate of the true distance. So don't forget to keep that principle in play regardless of how you're building a map. Okay. Well, I know it's a little bit much to take in, especially if you're not an expert in fungi, but now you are. So take some time to go over some problems with those, and I'll see you again real soon.